This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 487, recorded on March 30th, 2018. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Today we are in Milwaukee, Wisconsin. We are at the Medical College of Wisconsin. I have a sign right here that reminds me where I am, because just because it's Friday doesn't mean it's Milwaukee. And we're in the Department of Microbiology and Immunology. And we have with me today 5 individuals who are either here in the department right now or were here at one time. And we're going to chat with them about science, microbiology. And let me introduce them. First, we have a former chair of the department, a longtime secretary-treasurer of the American Society for Virology. In fact, since its inception in the early 1980s, Sid Grossberg. Welcome to TWiV.
Thank you. It's good to be here.
You know this, but perhaps the audience doesn't. I was the first Sid Grossberg lecturer. Is that right? Second. 1999. Why wasn't I the first? Never mind. It's okay. You don't have to answer that. Anyway, welcome. Great to have you. And next to Sid, and most of you can't see this because you're only listening, he's the current chair of the department. His claim to fame is that he was on TWiV number 40.
Wow.
Which was recorded at Cold Spring Harbor, New York. John Kirby, welcome to TWiV.
Thanks, Vincent. Good to see you again.
First TWiV and now TWiV.
Yeah, crossover.
So if you remember, that TWiV
Yes.
was at the occasion of the final phage course
right.
at Cold Spring Harbor.
Right.
And with you and Stan Malloy and Washlow Shibalski.
Yep, Václav Shibalski.
Did you know Washlow, Sid?
Very well.
He was quite a
University
character.
of Wisconsin.
Yep.
Yes.
And we did the episode in front of his portrait, which is in the Carnegie Library.
Exactly.
We sat him in front of it. It's a very majestic. He's standing next to a double helix. Also joining us, he's a professor in this department, Scott Terhune.
Thanks for having me.
And I call everyone professors. On TWiV, everyone gets a promotion. Everyone is a professor.
Did you hear that, John?
I did.
Thanks.
We should move to TWiV.
Wow, I didn't miss a beat there.
Also a professor in the department, Vera Tarakanova. Welcome.
Thanks for having me.
Did I say that right?
Kind of.
Tarakanova?
Tarakanova.
What?
Tarakanova.
Tarakanova.
There you
How do you say my name?
Racaniello.
Okay. That's
pretty
not
good.
bad. And finally, our final guest, last but not least, she is a AAAS Science and Technology Policy Fellow at the National Science Foundation and a former PhD student from this department, Dominique Carter.
Hi.
Welcome.
Thank you. Happy to be here.
Who I last saw in Canada
Yes.
at a American Society for Virology meeting.
Yes. And I have a selfie.
You have a
for this.
selfie. You have to share that.
I will. I am.
Okay. So we don't have a lot of time, but we'd like to find out some history of the department. And let's start with Sid. You were chair for many years.
Indeed, 31.
31 years. And before you were chair here, were you also a member of this department, or did you come from elsewhere?
I came very much from elsewhere. I came from New York City. At the New York Hospital, Cornell University Medical College.
Were you a faculty member there?
Yes, in microbiology, with an appointment in the Department of Medicine as well.
Okay. So, and tell us, where did you get, are you an MD or a PhD? I forgot.
MD.
Where did you get your MD degree?
At Emory University in Atlanta.
I'm going there on Monday to visit. Good. I'll mention you. Maybe they'll remember you. So you came here to be chairman.
Yes.
Why did you decide you wanted to be a chair?
Well, I wasn't quite sure, as a matter of fact. I was rather young. I was 36 years old, and I had contrary advice, although I had advice from former teachers who suggested that if I really wanted to do it, and they knew the institution, that they thought that it was an opportunity, but it would be entirely up to me. There was no virology in the institution at that time here. So it was a challenge. But one of the reasons for coming was the dean. Jerry Kerrigan was a Harvard-trained pediatrician and was determined, as a part of his vision, to build the basic science departments. And I saw lots of opportunities here. The parent institution originally was Marquette University, and the medical school was not doing so well under their administration. So it was actually fortunate that the medical college separated, and it has indeed flourished.
So you were able to build a department in your vision.
Yes. So it was, there was only one person in the department Wesley Hatlin, who had an NIH grant. And she was working on the genetics of Haemophilus. So it was difficult to recruit new faculty, certainly of the caliber that I had hoped for. But we managed, and I was able to build a research program for myself, and my intention was to build a research-oriented department. The department had a very good reputation among the students as being one of the best teaching departments. So that was fortunate, and we could build on that. I think we still are.
So looking back, would you say you accomplished most of what you had set out to?
Well, I think the way that the department has flourished, that it's more than I could have imagined. So I came to virology, though, in a curious way. When I was drafted into the Army, I out of a medical internship at Duke University, I was assigned to Korea. And I spent the next 8 months trying to get out of Korea. And fortunately, by great luck, got assigned to a research laboratory, actually the main laboratory for the United States Army and all the armed forces of the Far East in Tokyo. And as a part of that experience, it happened that I was in the circumstance to isolate the first Asian influenza, H2N2. It's A2 Japan 30557. It was the virus that got into they selected at Walter Reed, they selected that to be the vaccine virus. So I became an influenzologist.
Must have known Ed Kilborn then, right?
Indeed. Very well.
I have a feeling, Sid, you need a TWiV of your own. Because you probably have a lot of stories to tell that we don't have time to do today. But, I'm coming back to Wisconsin in December. You want to get together and just do a one-on-one TWiV?
that would be great. Just the story of the isolation
Would be cool. Yeah, love to hear it.
on an aircraft carrier.
Do the TWiV on the aircraft carrier.
Oh yeah, right. That'll work. So, John, could we get a room in December to do a TWiV with Sid?
Yes.
You can see it. That we'll find. Okay, that would be great. So if you look back on all the science you did, is there any one project that you particularly liked?
Again, because of circumstances and the phenomenon of viral interference, which was solved by Alec Isaacs in London as being due to interferon. A circumstance that actually fit in with the research project that I was assigned actually when I went as a fellow at Johns Hopkins, and we ended up by showing that viral interference at a distant site was due to circulating interferon in chicken embryos. So then had the opportunity, thanks to the NIH, to work with Andrei Lvov, the Nobel laureate, who discovered lysogeny in bacterial viruses. But he was working on polio at the time, a virus in which you are one of the world's experts. Oh, I can say it. And so at the end of that year is when he won the prize with François Jacob and Jacques Monod. So that probably helped my CV. And so it was a circumstance that an assistant professor was offered the chair here, assistant professor at Cornell. But even so.
Yeah.
So.
The people who are here, this is an amazing resource of virology history. There are not many like this, and that's why I would love to capture his story. And I hope you appreciate, when he says things like lysogeny, that was what was so cool back then, right? Not just...
Indeed.
Yeah. I'm sorry, I interrupted you. Go ahead.
So the department got running pretty well, but after about a decade, I decided, after working on establishing, it was a problem, the science in interferons, because it had this protein had such a high specific activity, it was very difficult to measure. And that whole mechanism, actually, it took almost 3 decades, eventually, for Jim Darnell at the Rockefeller to work out that whole multiplier system after gene induction. The problem in establishing ways to measure interferon became very important. And so one of the ways was to establish standards. And so in this department, we established the interferon standards for human interferon, the 3 types known at the time, alpha, beta, and gamma, as well as mouse interferon, the major working model. Subsequently, after that, we established antibody standards, and these became World Health Organization standards, most of them that we prepared for the NIH. But after all that, I felt that I needed my batteries to be recharged. And so I asked our then dean if I could take a sabbatical. And he said, sure. So I got a European molecular biology fellowship and began to work with Luc Montagnier. But Luc at the time had not yet invented HIV. So that didn't hurt my CV either.
It was Bob Gallo who invented it.
I'm sure that's what he would say.
Now, you said there won't be any editing to this conversation?
No, we'll leave that in. But Bob doesn't listen, so it doesn't matter.
So, and you've talked about ASV, which was the central nervous system here in the department, actually.
That's the American Society for Virology, not avian sarcoma virus, right?
No, that deserved its own Nobel Prize.
Right. It did several, actually.
That's true.
3. Rouse,
Yes.
Baltimore Temin, and Bishop Varmus. 3 Nobel Prizes for Rouse sarcoma virus.
That's true.
Amazing. So one last question
Yes,
for
sir.
now. If you hadn't been a scientist, what would you have done with your life?
I probably would have gone into the practice of neurology or the teaching of neurology, because that was the kind of research that I participated in as a medical student, and then in part during my internship, because it was a circumstance that happened to develop.
So after you stepped down as chair, then we had Paula Trachtman, right? Who was a friend of mine from the Baltimore lab. And then recently John is the chair. That's the line of succession, right?
It would appear, yes.
Now, when you were on TWiV, you were not here yet, correct?
That is correct.
So what enticed you to come here and be chair of this department?
That's a great question. And my answer is actually amazingly similar to Sid's. I came here because of the opportunity and because of the dean. I was not looking for a chair position. I was considering other potential options and wanted a certain institution with particular resources. And I ended up on a list of candidates to come here and interview. And of course, I had known many members of the department here at MCW and knew it was a strong department. And I learned about the history of the department and then came here and interviewed, and it was rather extraordinary. I was impressed with the agenda that is in place, that's ongoing, the building of new structures, the building of new centers, various elements of this enterprise. I think there's a really great vision coming from the president and the provost and the dean. They're committing their resources. They're basically putting their money where their mouth is. They really want to do something about it. And the charter that I have coming in is to essentially double the size of the department. And in so doing, we're going to renovate our entire basic science building and make that possible. So they have committed substantial resources to this. Things that are related to microbiology and immunology directly, we're getting a new center for immunology. We have a center for infectious disease research. We have a genome sciences and precision medicine center, and many elements are all being integrated, and we're hiring and committing resources to that. So
you've been here just over a year?
Yeah, 1.3 years instead of 31 years. Yes.
How would you say it's going so far?
Well, it's a challenge. They say it's like drinking from a fire hose, and then when you think you've handled that one, you turn around and there's another one coming at you. It's impressive. I still like it a lot. I actually love it. So I think that's a good sign from my point of view. I don't know what the rest of the crew thinks, but that's okay. I really like it. It's an unbelievable challenge. I have to represent the interests of the faculty to the rest of the university, and I have to represent the university's interests, excuse me, to the faculty. So that can be quite challenging because I get to impart, edicts and things like that, which aren't necessarily a favorable piece of information. But it's a great thing. I'm really quite happy with it.
Going to be here 35 years also, you think?
Well, that's an interesting thing that's totally unique about this. Every place I've been, the chair positions are either rotating or they don't necessarily come with, robust packages to do a lot. It can cost a lot of money to bring in someone from the outside and commit those resources. So it is an interesting opportunity that we can stay around long enough to see some of these things being implemented come to fruition. So that's a real positive. That was a real plus for me. So that's why I decided to do it.
And when I talked with you on TWiV 40, you talked about myxobacteria. Remember that?
I do.
But I understand you're doing some other interesting things. So what's turning you on in the lab right now? You can't. I know you got people here.
It's
If you say one thing, but they'll understand.
Yes. Well, it is the case that, so we're doing microbiome work right now, and we're very interested in phages, the lysogeny, lytic switch, et cetera. We're looking at phages and bacteria in the context of the microbiome, in the context of animal guts or the human gut. But the perspective that we take on that is 100% learned from what we learned looking at the soil bacteria. So our myxobacterial projects led us to the path of looking at predator-prey dynamics. So we're still doing that. We're looking at Myxococcus xanthus, Bacillus subtilis dynamics. They use essentially chemical jousting. They're each producing these small molecules, which to us would look like antibiotics. They're made by polyketide synthase clusters or non-ribosomal peptide synthase clusters. We can purify those molecules. We can add those to to these organisms in vitro, do plate assays, and we can figure out the rules that govern those interactions. So then we actually, from going to Cold Spring Harbor, teaching the advanced bacterial genetics course, back in 2006, I think it was, I heard Forrest Rohr talking about the new white paper for the Human Microbiome Project coming out of NIH, and there was already an indication for wanting to look at phages there. And having come from the University of Illinois and Urbana-Champaign, I've been saturated with discussions on phage biology, so that had been on my radar screen for decades as a student, since I was a student. And so what we started doing was thinking about experiments that would interconnect these small metabolites, these antibiotic-like molecules, the role of phage biology, and the predator-prey dynamics that we're seeing in the soil bacteria. We actually published a 2006 paper, and I think it's Journal of Bacteriology, where we looked at how a lysogen in myxobacteria disrupts its development, its multicellular development. So we've known from work in our lab that phage affect multicellularity. So we started doing these experiments based on what I'd learned from listening to Forrest and a few other people. We started doing experiments to get going with microbiome research. And I finally recruited a graduate student to do a rotation in, I think, 2009, when I was still at the University of Iowa. And we got the microbiome project up and running full gear. And the luck of the draw there was that we ran into a psychiatrist who was interested in understanding why treating patients with drugs was leading to obesity and type 2 diabetes. And so we then picked up that and ran with it. And the molecule of interest is risperidone, which is one of the most widely used antipsychotics in Western Hemisphere in modern molecular medicine. And that molecule has these really nasty side effects. And so we have moved the experiments into mice. We treat mice with risperidone. We get major shifts in the bacterial population. There's corresponding shifts in the phage population. And we can even see now from our ability to do metagenomics and mine all of that data, we can see that there are polyketide synthase gene clusters that are differentially regulated in response to these small molecules. We even have an example that I don't want to discuss on the podcast because we're getting ready to file a disclosure. So we have a small molecule that protects against the xenobiotic. Mm-hmm. So what we learned from the soil bacteria is now what we think is happening in the human gut.
Let me see if I get this. So something produced by phage lysis, induced by this drug, is releasing compounds that then have neurotropic activities?
Well, I would say I'm going to take a step back, and we're going to call it a quadripartite system. Mm-hmm. Small molecules and phages interact with the entire consortium to regulate the overall ecology of that microbial forest. And then that contributes to host health or disease. So the phages are not necessarily directly linked, or they don't necessarily carry the genes for polyketide synthesis, but they can they certainly co-occur or co-exclude. They may, in fact, carry some regulator genes that lead to production of those molecules.
Okay, so someday if we understand this, it may help understand how this drug works and maybe lead to even better ones. Right?
That's what we think. Yeah, we think that we'll be able to develop probiotic formulations, which would help stabilize an individual before they even begin treatment.
Yeah. So it's interesting because one of my areas of interest is chronic fatigue syndrome, and because a number of years ago a retrovirus was suggested to be involved, which turned out not to be the case, but it got a lot of patients following my writings and podcasts. And I got an email last week from someone who said, what if a phage lysed bacteria in the gut, which then released substances which got into the brain and were in part responsible for chronic fatigue? And I said, why not, right? Mm-hmm. What do you think?
Well, I think that's a great hypothesis. I can't remember the name of the individual who did the study, but quite some time ago, it was recognized that peptidoglycan fragments were a sleep factor for rats. Mm. It is and there's now a whole slew of studies that have come out that have said that circadian rhythms are being coordinated by or co-regulated with bacterial flora. Mm-hmm. Right. So I think that's quite likely to be the case. It's circadian rhythms is a hot issue in the microbiome field right now.
It just got a Nobel Prize last year. Yes. Clocks, right?
Clocks, that's right.
So in a few years, you'll be on TWiV talking about this, right?
I hope so.
All right. So you could be a chair and have a robust research program. Sid showed that years ago. You can still do that nowadays?
Oh, it's incredibly difficult. I'm 100% chair and then part-time scientist. I have to depend on the people in my lab to really carry a fairly heavy load. I've got great research scientists and postdocs helping manage what's going on in the lab, and the students are very independent-minded, and they're doing a lot. So yeah, you can pat yourself on the back over there.
Do you think you'd say that if you weren't here?
I would have, yes. Yeah. No, it's definitely that's a challenge. The first year was me completely learning the job, and now I'm able to do more work with the students. I've had to change my schedules a bit so I can accommodate everything.
I was just at David Baltimore's 80th birthday celebration, and he had, as part of a full-day symposium, people in leadership positions that had come from his lab, like the president of the University of Michigan, the dean of Harvard Medical School, you get the idea, right?
Right.
And each and every one of them said, it's important to have a lab when you're in these positions because you remain in touch with the science. So no matter where it is, chair of a department, a dean, even a university president, apparently Schlissel has a lab waiting for him, should he want to use it. It's important to stay in touch. Absolutely. And not just stop research and And you don't know what's going on.
Right.
All right.
It does require staying up late.
It does. Yeah. I stay here later, so I have most of my meetings up till 5 p.m., and now I work with the students like between 5 p.m. and 7 p.m.
Is that right? Is he telling the truth? Okay, good. Not that I would doubt you, John.
Well, we have a meeting coming up later this evening.
Let's talk with Vera a bit. Where are you from originally?
I'm from Minsk, Belarus.
Belarus.
Yes,
Well, you say it properly. Okay, Minsk.
Minsk. Yep.
And how far in your schooling did you do over there?
I actually went all the way through high school.
Okay.
And then I was a participant. So at that time, which reveals my age, Soviet Union was falling apart. And the U.S. government thought it would be a great idea to fund a program where they would bring, potential leaders to the U.S., imbibe them for a year, have them stay with the American families, go to high schools here, and imbibe them for a year with the principle of democracy and other propaganda that they could then take back to the former Soviet Union and implement. And so I did that for a year, but I think their fatal mistake and kind of a not a very clever use of the taxpayer dollars was that the people they selected, high school children, were not that dumb. And after being in the U.S. for a year, they decided it was a great place to live. And so, as far as I know, most of the people who participated in this program are now happily living in the United States and running their careers here. So then I came back to go to college, and that was it.
Where did you go to college?
I actually graduated from University of Tennessee, Memphis. I was going to be a medical technologist. And then I decided it was very boring.
Mm-hmm. And so you decided to get a PhD?
It seemed like I wasn't really going into graduate school to get a PhD per se, because I wouldn't know at that time what to do with it. I just wanted to play, and it seemed like a perfect opportunity to play, and somebody was actually going to pay me for it, however little money. So it was sort of irresistible at that time to do such.
And where was that? Where did you go from?
St. Louis University.
Bill Wold, right?
That's right.
As I learned earlier.
Yeah.
That's where you got introduced to viruses.
Exactly. I always liked microbiology, so I worked, even when I was in the hospital, I much preferred the microbiology department. So I had quite a bit of clinical bacterial experience, but virology wasn't something that we did in that particular hospital. And so I ended up rotating in Bill's lab because I wanted to see what viruses were like, and that was it. That was the beginning of a lifelong affection.
Viruses tend to do that.
Exactly. They tend to hang out with you for a very long time, too.
And then you stayed in St. Louis for a postdoc, correct? With whom?
I went to Skip Virgin's lab.
And which virus did you work on there?
So I wanted to now, have a system where I could play not just in tissue culture, but I could have animals I could infect. And I also like cancer-associated viruses, so mouse gamma herpes virus system was quite perfect for me at that time.
So gamma herpes virus 68, right?
Correct.
Which is a mouse virus, not a human virus.
No, it was actually isolated originally from a bank vole in former Czechoslovakia in the late 70s. But turns out it's actually present in rodent populations, in feral rodent populations. So it's a pretty promiscuous virus for rodents.
Why is it a good model? What does it tell us?
So it allows us to do experiments we would never be able to do with human viruses that are exquisitely human-specific. So there are, many, many elegant studies that have happened over decades, dissecting out various components of biology and virus-host interactions, say for human EBV and KSHV. But the problem is it's very difficult to verify if those interactions are indeed happening in vivo. And many ways you can design, many targets, therapeutic targets you can design based on in vitro work don't actually pan out when you go in vivo. So the beauty of the system is, to me, at least we can, yes, identify new targets, and we can narrow down the list by doing the entire spectrum of studies to something that we know works, as I tell my students, something that works in a thing with 4 legs that runs around and poops. And so, to me, that's really the value of the system. It allows us to focus in on things that are important.
So you came here from Skip's lab, right?
Correct.
And brought Gamma 68 with you, and you continue to work on that?
Yep.
So what is exciting you in the lab with that virus these days?
Yeah, so, the beautiful thing about viruses is you never know what you're going to work on. Because if you listen to the virus will give you a direction of where to go, and that may not be the direction that you wanted to go initially. So we've gotten fascinated with interferon regulatory factor 1, or IRF1, and it's very intriguing protein that has been sort of orphaned for a number of years. It was the first one in the family to be discovered because it binds to interferon beta promoter. So people were very excited because they thought this was going to be the host factor that drives expression of this very important host antiviral molecule. And it turned out that it really doesn't. Even though it binds to the promoter, people soon discovered that there were other IRFs, like IRFs 3 and 7, that are much more important. However, this transcription factor can restrict replication of almost every virus that's been screened so far. And it's clearly not through induction of interferon. So that's very interesting. But another exciting and unexpected thing we've discovered, when my former student put the virus into IRF1 deficient mice, which of course goes back to working with the whole continuum, we got this really interesting phenotype in the spleen with latent virus. And so to make a long story short, what these clever viruses do, both human and mouse viruses, they infect B cells, which is sort of weird for most viruses. Most viruses don't do it at all, or they don't do it very often. But these guys actually depend on B cell infection in order to establish lifelong reservoir in a host. And so when these viruses infect, they drive differentiation of B cells through the stage called germinal center response. And it's a very weird germinal center response because normally only B cells that are specific, say, for a viral antigen have the business of being there. But with these viruses, they actually drive all kinds of B cells with specificities against self-antigens and even antigen of other species to go through this differentiation process, however briefly. And so what we unexpectedly discovered was that this IRF1 transcription factor restricted gamma herpes virus-driven germinal center response. And so when we first saw it, we saw that, well, whoop-de-doo, we discovered the homeostatic factor of the host that restricts any germinal center response. But this was not the case when we infected mice, IRF1 deficient mice with the unrelated virus like LCMV, or simply immunized them. Their germinal center responses were normal, suggesting that IRF1 selectively restricts germinal center response driven by this virus. And what's important about it is that, remember, I told you IRF1 is also a tumor suppressor. And germinal center B cells are what's thought to be targeted by EBV, and perhaps even mouse virus, even though we haven't looked at it. They're targeted by EBV for malignant transformation. And so we may have found a tumor suppressor for virus-driven lymphomas, although this is very much an overstatement, and I have to classify it like that. However, when we did look in EBV-driven lymphomas that arise in transplant patients due to a very strong transplant program here at MCW, what we found was that IRF1 expression in B-cell lymphomas that are driven by EBV is selectively lower than in B-cell lymphomas that are EBV negative, suggesting at least correlationally that we might be on the right track. So we're following this up now with some animal studies and mechanistic studies.
So when you infect mice with gamma 68, it preferentially goes to lymph node germinal centers, is that right?
That's correct. And that was shown years ago.
And it doesn't replicate lytically in the B cells, it establishes a latent infection.
That's right. So these clever viruses manipulate the host so that if they can go latent in germinal center B cells, which are thought to divide about every 8 hours, so they're almost like bacteria, the virus can stay latent in those cells and have its latent reservoir being expanded exponentially just due to cell division, which is a very brilliant and sort of evil strategy of the virus.
And that's what IRF1 is blocking.
Yes.
And the mechanism is something you need to find out, right?
Well, hopefully one TWiV, we can talk about that in the future.
Yeah, but that's a question, of course, because if IRF1 is restricting proliferation in some way, it would be really interesting. Because you said it was a tumor suppressor, right?
Yes, also in ways that people don't understand.
Yeah. Right, cool. Now, if you could give any advice to a young person who wants to do what you have done, what would you tell them?
I think the most important thing is to have fun. You have to find an area that truly excites you. And if you're having fun with that, you are motivated to do your best work possible and to do a lot of it. And so I think that's really a prerequisite for success in science.
I've heard her say fun more times today than any other word. Unusual for a scientist to say that. Scott, where are you from?
I'm originally from Minneapolis and did my graduate work at Northwestern on papillomaviruses.
Lou Lehman?
With Lou Lehman's. But actually, I caught the virus infection as actually an undergrad. I was able to do a research project with Pat Spear at Northwestern on herpes simplex virus entry. And as soon as I started learning about viruses, I was hooked. I mean, viruses are just extraordinary, simplistic pathogens that have very, very sophisticated mechanisms to control cells, to control host systems, and so that's really what kind of drives me. And I would agree with Vera. I mean, you find that one thing that you find really amazing, and it is a lot of fun. Certainly there's times where it's challenging, but overall it's, that's what drives, at least what drives me is just this interest in viruses.
So you let the viruses lead you. You don't lead them, right?
That's right.
So, I'm sorry, you're a PhD with Lou? Is that right?
So I did my PhD with Lou,
right.
And I, thought, studying papillomavirus, it was very exciting, but it was a virus with about 8 open reading frames. So it was a DNA virus, a small DNA virus. So then I went into my PhD work in Tom Shank's lab and moved back to herpes viruses and studying cytomegalovirus. And it was a great time because the back recombinating methods were just being introduced, which made studying CMV a lot easier. So in Tom's lab, I learned back recombinating methods, and in parallel to that, started learning peptide mass spectrometry in collaboration with Brian Chait's lab. So that combination is really what established my career. And that's what we continue to work on, which is understanding viral protein function through making a mutation in the virus, evaluating the phenotypical outcome, and then identifying in the wild type virus who that viral protein is interacting with, leading to, some fantastic hypotheses. So again, I mean, we just followed the discoveries, and it sometimes brings us down some roads that are a little bit more challenging than others, but it is a lot of fun, and I've really enjoyed it. And
so you came here from Tom's lab, is that right?
I came here from Tom's lab and started in 2007 under Paula Trackman, right. And I have to say I started my assistant professor position when I was in my late 30s. Sid started his chairmanship when he was 36. So I'm doing my best, Sid. I'm doing my best.
You're doing very well.
Yeah.
John, you're not 36, right?
No.
So what is exciting you these days in the lab?
So over the years, we've identified and defined some very interesting molecular mechanisms, and we continue to do that. Each student comes in, will identify a protein they're interested in working on. We figure out kind of the molecular mechanisms of its action. But over the years, we've been seeing some kind of central trends, and a lot of the mechanisms are overlapping. And one of the areas that we've been studying more recently is how the virus is controlling the mitotic cell cycle machinery. That's important because some of the antivirals against CMV utilize that machinery. It's part of the antiviral activity. We've been moving into taking some of those molecular mechanisms and working with mathematicians to start to define those relationships based on mathematics. And I'm very excited about this. It's every day is really hard when I'm working with them because we really speak different languages, and I have to explain molecular processes, and they explain mathematics, and they'll model something that we can't experimentally determine. So I have to go back and have a conversation of, we just can't determine that. You'll have to switch things around a little bit. So we're moving in, taking a lot of this molecular mechanism and hopefully develop some great kind of predictive models, focusing again on mitosis and the oscillations in mitosis. So I'm really excited about moving forward.
So is it the case that the virus needs to control the mitotic cycle to be able to utilize DNA synthesis apparatus? Is that the main reason?
Well, the main reason, so the virus shuts down the host DNA synthesis machinery, but it still utilizes the rest of cell cycle machinery in the process of essentially assembly and egress.
Okay.
And so it pushes the cell into mitosis, but it needs to prevent it from finishing mitosis.
Right.
So there's a push into mitosis and then an initiation of degradation of some of the machinery that prevents it from leaving mitosis.
Right.
And one of the kind of final stages is the virus is mimicking nuclear envelope breakdown in order for the virus to get out of the cell. And a lot of that mechanism was actually done by Don Cohen at Harvard, but it fits into a lot of the mechanisms that we've been studying over the last few years. So it's really quite sophisticated. It's mimicking some of the processes just to create an environment where it can assemble and establish egress.
So as you know, HCMV is a major cause of congenital birth defects. And back when Zika emerged, I was speaking to journalists about it, and I said many times, this is not the first virus that is causing birth defects. And I said, for example, human cytomegalovirus, and they had no idea. How about rubella? They had no idea.
Yeah, it's really interesting. The media really put Zika virus on the spotlight, and a lot of these, pathogens that have been around forever that cause congenital birth defects are still causing congenital birth defects, are still the leading cause of congenital birth defects.
Absolutely.
And Zika, it's still out there, it's still an issue, but it's not as big of an issue.
I mean, it's not to minimize the importance of it, but there are already others, and to think that that's something novel is just wrong. And I think that's our failing as scientists. We haven't educated the public sufficiently, and that's what we try and do with these podcasts. And unfortunately, science writers don't listen to them. But future science writers do, and so maybe we'll get them. So if you had to give advice to some new person who wanted to follow in your footsteps, what would you tell them?
Well, again, you have to follow what you're passionate about. It's a difficult job. There's certainly days that are challenging, but it's really the excitement of the career that keeps me going in the topic. So that's what students need to identify. And then one other thing that I often focus on, at least in terms of training my students, is to teach them how to be great scientists, to think about, formulating, solid hypotheses, testing those hypotheses, but also trying to provide some sort of unique kind of a skill set that they can take with them and be a great scientist, but also be very unique in that environment. So Dominique Carter is...
Did you work with this fellow?
Yes, I did. He was my former mentor.
And when did you finish?
I finished in 2016.
Okay, not too long ago. So you decided not to do bench science. When did that come to you that you didn't want to do that? Do you remember?
I think it came to me during probably the height of the funding crisis. And seeing colleagues or former colleagues worry about what their transition was going to be like in academia and, what opportunities awaited them. So I started looking into other areas that I would be able to use, what I was trained to do, which was solve a problem at the end of the day, ask the relevant question and come up with a sound approach and mechanism to solve it, or at least a part of it. And I became interested in science policy more so kind of because I was curious. How can this be, right? Science and engineering research drives innovation. And, America was once preeminent, in scientific discovery. And how has it come to a point now where, up and coming scientists have to worry about if they can maintain or, stay in this type of, have a successful career in academia. And that's what propelled my interest at first. And so from that, I started asking questions. Scott was gracious enough to kind of allow me toward the end of my graduate school tenure to start finding an answer, or at least what was going on that led to this.
I did try to convince her to do a postdoc in maybe a higher profile lab on one of the coasts, but
He did.
she didn't listen.
And so even now, I very much enjoy what I do, but I will tell you, I do miss the lab some days. And I often worry about kind of how to tread this line of, doing this important work in policy, right, at the intersection of science and policy and innovation and, basically having scientists and engineers and physicians engaged in policy, to promote data-driven policy making and decision making with regards to our fields and, the things that keep our lights on at the end of the day. However, I still recognize how important it is, to stay in a lab and how important it is to provide opportunities for up and coming scientists to stay in the lab, right? So yeah, I often think about it.
So what do you do as a Science and Technology Policy Fellow? What does it entail?
So, AAAS fellows serve in the 3 branches of government: executive, legislative, and judicial. I'm in the executive branch, and I'm a fellow at the National Science Foundation, the largest funder of basic science research since its inception in 1950. I'm housed in the Office of the Director, and specifically within that office, I am in the Office of International Science and Engineering. So my work is mainly focused on science diplomacy and policy. And so it's an interesting time to be working in that right now.
But what do you actually do on a day-to-day basis? Who do you talk to? Do you write? Do you visit?
So I write, I talk, I talk to people across the foundation. So the foundation is organized by 7 research directorates, having a specific scientific discipline except one, which is called the Education and Human Resources Directorate. And that's focused on, revamping and science curriculum and STEM education. And so in that office right now, for policy work, I'll say I am working with 2 program officers to co-author a solicitation. And this solicitation will be focused on supporting international research networks of networks. So what that is, the landscape of science is changing, right? As globalization is becoming larger, science is becoming more global. We actually released in our National Science and Engineering Indicators report that for the first time in history, the U.S. has been eclipsed by China in terms of overall federal investment in science and engineering research. And so for this solicitation, it's specifically focused on creating how to support the creation and maintenance and management of international research networks of networks. So networks of scientists collaborating with the networks of scientists across disciplines in multiple countries.
Mm-hmm. Can we reverse this trend of losing dominance in science?
We can. Let me not get ahead of myself. The optimistic Dominique would say, yes, we can. But the realistic version would say that we can slow this process down. And how do we do that? By recognizing that the landscape of science is changing. And instead of using approach that is not as, inclusive and more exclusive and kind of focused on, how can America drive innovation? What is going to be good for America? What's going to be good for America is when, we invest and recognize the significance of working with other countries. So for example, right, as the U.S. has kind of divested, in biomedical or basic science research, just based on our history and overall, assigning a portion of or fraction of our GDP to support basic science research compared to smaller countries with a fraction of our GDP. So they literally have a fraction of our output coming out, but they're investing more of their GDP in science. And we're actually doing the reverse trend. So first, we need to stop that. I'm encouraged with this new omnibus budget bill that was passed. I'm sure if we could have some champagne in the government, we probably would have. Many people were extremely concerned because, all of the people at NSF that are at least in the program staff was once a faculty at one point in time, and they recognized the value and significance of supporting awesome research and good work. And so I'm encouraged by that, but we have a lot of work to do.
How did that come about? Because the administration is decidedly anti-science.
Mm-hmm. Yes. So, good question. It came about from the National Science and Engineering Indicators Report, which is a congressionally mandated report that's required to come from the National Center for Statistics and ES, I don't know what the ES stands for, NCSES, which is housed within the Social Behavioral and Economics Division or Directorate at NSF, to kind of look at the trends, global trends and domestic trends in science and engineering research. And that report showed, like I stated earlier, that the U.S. had been eclipsed in overall federal investment in science. And if people don't understand, basic science research or why it's important, they understand numbers at the end of the day. And now, we are now, not the largest producer of publications anymore. China is. We are not the largest investor in terms of investing in science and technology startups. China is. We are no longer, well, I can't say that. We still are the most cited. So the U.S. still has the most cited publications. We do have that. But just in terms of overall investment, the U.S. is losing. And so, Congress was like, well, we can't have that. And so they totally went in the opposite direction from what all trends and what was said by, the Science and House Committee, like what they were going to do, went in the opposite direction, which was a pleasant surprise for us.
Sure.
Right.
So we could talk a long time on this. In fact, are you in Washington? Is that?
Yeah, I'm not in Washington proper. I'm in Alexandria, Virginia, but yes, Washington, D.C. metro area. So
I'm at ASM in D.C., and sometime we have to do another podcast and get more deeply into this, because we could talk about policy and science for a long time.
Absolutely.
But we don't have a lot of time now. But our last administration was completely pro-science. Wonderful.
Oh, yes.
Right? The Office of Science and Technology Policy was well-staffed. And do you interact with that office now at all, or?
Not as much because, a science advisor has not been appointed.
That's
yet.
right.
So many of the offices that do not have leadership are trying their best to maintain, but without leadership, it's hard to determine direction.
Sure.
Right. So
you're trying to be careful, I can see.
And that's something challenging for me because I'm a straight shooter. And so that's something that I've learned.
Well, you can be sure that no one in the executive branch is going to listen to this podcast.
You never know.
I assure you. Although we used to have Jo Handelsman on TWiV before she went to OST, which she did for the Obama administration, which was great. So let's do a podcast sometime in the future and talk about it
Absolutely.
in more detail. But for now, one more question for you. Is what you do really rare, or can anybody here think about a similar career? And if so, how could they do it?
Anybody here can think about a similar career. If you're interested, you don't have to wait to do a fellowship like this, like the AAAS Science and Technology Policy Fellowship. You can get involved at a local level. That's what I did, and Scott encouraged me to do so. I did some work with Wisconsin State Senator based on STEM education of underserved communities here in Milwaukee. I looked for other opportunities, for example, professional societies. Many of them that are in our disciplines sponsor like a Capitol Hill Day where you can go and advocate for science or on behalf of your society for a couple days, visit the Hill, visit, your state legislature, state senator. I did that. Yeah. So there are a lot of ways you can do it.
So your fellowship is for a defined period of time, right?
Yes.
And then what do you do next?
Who knows what? No. So, so first of all, going into this fellowship, it's early career, mid-level, senior level. Like somebody in our class was a professor emeritus, and he just wanted to work on the Hill. But people go back into academia, people go into private sector, some people go back into nonprofit, and some people go and work for the government. I don't know how this is going to work with this class, but we'll see what happens in 2 years.
Great. All right. That is TWiV 487. You can find it at microbe.tv/TWiV. If you have a cell phone, mine happens to have TWiV on the back.
Oh, cute.
But if yours doesn't, it will still play TWiV. You can just go to your podcast player and search for TWiV and subscribe so you get every episode. And we love getting questions and comments from listeners. Send them to twiv@microbe.tv. And of course, if you love what we do, you can support us financially because all of us do this for free. You can go to microbe.tv/contribute. My guests today, all here or formerly from the Department of Microbiology and Immunology at the Medical College of Wisconsin. Scott Terhune, thanks for joining us.
Thanks for having me.
Dominique Carter, thank you very much.
Thank you.
Sid Grossberg, thank you.
My pleasure.
John Kirby, thank you.
Good to see you again.
And John invited me originally, so thank you for having me here.
This is great.
And finally, Vera Tarakanova.
Good enough.
Thank you very much.
Thank you.
Thank you.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the American Society for Microbiology for their support of TWiV. And Ronald Jenkins, that's the music you hear at the beginning, ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.